Hello everyone, welcome back to your classes on overview and integration of cellular metabolism. From today's class, we will be starting heme metabolism. Specifically, today we will be discussing about heme synthesis and regulation. So, you might wonder why we are studying heme. This should raise your question. Whenever you see a topic, you must ask why. Why we will be studying the structure of porphyrin and heme in this class? Why do we need to need to know the pathway of biosynthesis of heme? Why do we at all need to know the regulation of heme synthesis? This is because most of you already know that this has got a big role to play in red blood cells. Okay, so when we are discussing RBCs, you should know first a basic thing about RBCs, a very basic knowledge that RBCs are about a biconcave non-nucleated structure which have got 7 micron, right, structure, and their half life is 120 days. This you already know from discussion regarding HbA1c, that RBC of lifespan is 120 days, and that is why it has got some importance, right. So, this mature RBC do not have nuclei and hence they do not undergo the pathways that are there inside organelles like mitochondria and nucleus. However, RBCs do have cytoplasm and they do have the cytoplasmic enzymes. So, they mainly depend on glycolysis for energy and production of 2,3-BPG. This is all discussed, these are just revision lessons about RBC, right. So, you can see unknowingly you know so much already about RBC, and you also know that the NADPH that is required in RBC to fight off those culprits that were trying to degrade the RBC membrane, they come from the HMP shunt pathway, right. So, what is the main role of RBC? Why do they look red? They mainly carry hemoglobin, and for that heme synthesis is one of the main role of red blood cells, right. They form, you already know, they form they are formed in the bone marrow, and this reaction needs many compounds or many elements, many nutritional support. They are some amino acids, they are iron, copper, vitamin in the form of folic acid, vitamin C, vitamin B12 and vitamin B6 and pantothenic acid. So, all of these are needed for formation of blood and they are known as hematinics. So, whenever we utter the term hematinic, it means any drugs that are prescribed to form blood, okay. And these are specifically prescribed during pregnancy. So, blood forming syrups, all these things contain not only iron, but multiple, all of these are mixed, okay. So, with this basic knowledge of RBC, we should also know some basic knowledge about the structure, we should also have some basic knowledge about the structure of hemoglobin. We already know it is an iron containing conjugated protein that has got heme and globin. So, heme is the actual prosthetic group and the protein is the globulin. If you look at the structure, it is a tetrameric protein, 4 subunits are present in each subunit having a prosthetic heme group and the globin polypeptide. 2 alpha and 2 beta forms the 4 tetramers, okay. The molecular weight is 67 kd, kilodalton, or 67,000 Dalton, right. And each gram of hemoglobin, mind it, hemoglobin unit is in gram per deciliter, they contain 3.4 mg of iron, ठीक है ना. So, why do we need to know so much about heme? Because heme is not only present in hemoglobin, it is also present in myoglobin, cytochrome, peroxidase, enzymes like peroxidase, catalase, tryptophan pyrrolase and nitric oxide synthase, okay. So, very important, these enzymes also contain heme, and you may get a multiple choice question on MCQ where each of these may be given as an answer which you can choose as a direct answer or as an all except type of question. So, first things first, heme is produced by the combination of iron with a porphyrin ring. It is similar in which the counterpart, that is chlorophyll in plant, is also a combination of a magnesium ion and a porphyrin complex, right. And you know, the scientist who actually received Nobel Prize for his work on hemine and chlorophyll is actually Hans Fischer. He was a German scientist who discovered the role of both chlorophyll and hemine. You know, he was a so much dedicated scientist that he dedicated his whole life into finding all of this, and his whole life, entire work in Germany was actually destroyed during the end days of World War II, and he committed suicide. It's a very sad story, but it is a very inspiring that he already got Nobel Prize. Very vital in this context of our discussion. So, regarding the structure of heme, it is a derivative of porphyrin. Porphyrins are cyclical compounds formed by fusion of 4 pyrrole ring. What is pyrrole ring? This is how a pyrrole ring looks like, and when we abbreviate it by omitting the carbon and hydrogen, it is often signaled as this, right. And these 4 pyrrole rings are linked by methynyl bridges, and since iron atom is present, it is also a type of ferroprotoporphyrin. Okay, heme is a ferroprotoporphyrin. We will be discussing what we mean by protoporphyrin. Okay. So, then regarding the nomenclature of pyrrole rings, they are named as alpha, beta, gamma and delta, okay, based on the sequence. So, this is alpha, beta, gamma and delta, and generally we often I mean, for simplicity of understanding, we often represent this as in this structure, okay, where the sites for attachment of side chains are represented by the corner of this huge plus sign. Okay. So, you should get used to this type of nomenclature, but in reality you should have in mind that in reality that is 4 pyrrole structure which are connected by methynyl bridges. So, the porphyrin ring, this is the porphyrin ring, okay. This porphyrin ring are of multiple types, multiple subclasses. The first one, that is 1, okay, they have got an arrangement, symmetrical arrangement of substituent groups. We will be seeing what are the substituent groups very soon. So, number, if you just number it like this way, right. So, if you just notice the number of groups, the groups that are present in 1, 3, 5 and 7 and 2, 4, 6, 8, if they are symmetrical, then it belongs to first series. If they are asymmetrical, then it is known as series 3, right. And type 3 is the most predominant variety in biological system. There are also multiple types, but we do not need to know that. If you just know type 1 and most importantly 3, you are good to go. It is also called series 9, right, because Fischer, the discoverer, the pioneer in this porphyrin chemistry, has placed as the 9th series in 15 possible isomers. Okay. So, if we look at all the possibility, the original discoverer placed this, our product of importance, in 9th. Okay. So, it is also referred as 9 in some textbooks, okay, to respect the discoverer, but you should always know type 3 is the most predominant and this is the most interest for us. Okay. So, what are the substituent groups? What groups can be placed in 1 to 8? Okay, these are the ones: propionyl, acetyl, methyl and vinyl. They are often abbreviated as PA, V and M. We will see very soon, right. So, depending on the order of substitution from 1 to 8, this is the nomenclature, and there can be again multiple isomers or multiple forms of this porphyrin. To us, for heme synthesis, these are the intermediates that are required and these are the substitutions. Again, it is almost an impossible task to remember, but you still need to remember few which I will tell you very soon. So, this is the structure of heme, in which you should remember the substitution in order to get some good exam. This is a nice to know area, it is not a must know area, but you should know, right. So, in heme the substituent groups are methyl vinyl, methyl vinyl, methyl propionyl, propionyl methyl. You can remember it in any way, you can remember MV, MV, MPPM, it is up to you, but you should remember the final structure of heme and its substituent groups, right. So, heme is actually can be synthesized in almost all tissues in the body, right. In case of RBC, it is synthesized in not the mature ones, but the immature in bone marrow, that is in the normoblast. It is actually the intermediate normoblast where hemoglobin first appear. You already might be knowing that from your physiological knowledge, right. And if you look at the pathway is partly happening in the cytoplasm and partly in the mitochondria, and this is the reason why when RBCs are fully matured, they do not have mitochondria and heme synthesis cannot take place because there are few mitochondrial enzymes that are also essential in heme synthesis. So, this is the step 1 we have already discussed during glycine metabolism, how heme was being synthesized as an intermediate. So, succinyl CoA and glycine under the action of the enzyme delta aminolevulinate synthase, it is forming delta aminolevulinate. There is a ketoadipate intermediate, but ultimately this is the final reaction, right. So, succinyl CoA glycine forming ALA, this is also abbreviated as ALA or ALA. One very important thing to note is this requires pyridoxal phosphate as coenzyme, and this is the reason why in vitamin B6 or pyridoxine deficiency there will be defect in heme synthesis and there will be anemia, okay. This is a rate limiting enzyme. If we control the activity of this enzyme, heme synthesis will be affected and regulated and controlled, right. This is the major pathway that is happening in higher animals, but in bacteria and plants an alternate source of ALA is also glutamate, right. It can be directly synthesized from glutamate via a semialdehyde form, okay. So, in the second step what is happening, 2 molecules of delta aminolevulinic acid are joining together, right, and it is acted upon by a dehydratase enzyme. This enzyme has got multiple names, we can call it ALA dehydratase depending on the action on the substrate, or we can also name that enzyme depending on the product it is forming, that is porphobilinogen synthase, it is the same thing and leads to the formation of porphobilinogen, okay. So, this is the second step in which 2 molecules, why it is showing 8 and 4, because in reality we need 4 molecules of porphobilinogen. So, actually at one reaction level 2 molecules of ALA are combining, 1 molecule of water is lost and porphobilinogen is formed, but in reality 8 molecules of ALA are forming 4 molecules of porphobilinogen. This is the step 3. So, what happens after formation of porphobilinogen? So, 4 molecules of porphobilinogen results in the formation of the first porphyrin of the pathway, that is uroporphyrinogen, okay. Uroporphyrinogen is a cyclical compound, this is not yet cyclical, okay. This is, you can see the enzyme uroporphyrinogen synthase, actually the 4 porphobilinogen combines in such a way so that a linear tetrapyrrole ring is formed, it is not yet cyclized, okay. This is known as hydroxymethyl bilane or pre-uroporphyrinogen, HMB. This enzyme is also often known as hydroxymethyl bilane synthase, HMB synthase, okay. We will be discussing in detail again in porphyria, so do not worry. So, uroporphyrinogen synthase, it forms a linear compound which undergoes spontaneous cyclization to form uroporphyrinogen 1. Ultimately that 1 is a symmetrical porphyrin, okay. What happens? By uroporphyrinogen co-synthase, it is converted to uroporphyrinogen 3. So, everything is happening in one step. So, this linear compound pre-uroporphyrinogen is being converted to uroporphyrinogen 1 spontaneously, and then by the action of uroporphyrinogen 3 co-synthase it is converted to uroporphyrinogen 3. So, uroporphyrinogen 1 is basically the symmetric isomer, okay, in which everything AP is present in 1 to 8 in symmetrical manner, and only thing that is different in uroporphyrinogen 1 and 3 is the interchange of this A and P. If it goes here, it becomes third isomer. So all this is all what is happening in this reaction. So ultimately we are getting uroporphyrinogen type 3. Okay. So what happens when this fusion occurs? There might be many 1 isomer, there may be multiple 3 isomers, okay, because not all the 1 are converted to 3, but from now only series 3 are further utilized. Okay. The pyrrole rings will be joined by methylene bridges, which is derived from the glycine. Okay. And we should know that porphyrinogen are actually colorless. Still we are in porphyrinogen. These compounds are named porphyrinogen, but once they are oxidized, we will be reading very soon, they are known as porphyrins, which are colored compounds. Okay. So in the next step, what happens? Uroporphyrinogen loses carbon dioxide to form coproporphyrinogen. Uroporphyrinogen 3 forms coproporphyrinogen. What actually happens? The acetate groups are decarboxylated to form methyl group. There are 4 acetate groups, CH3COOH. Basically CH2COH, the carbon dioxide, if it goes, it will form, it will, CH3 will remain, and that is exactly what is happening. It is very easy to understand, and molecular, okay, you should know molecular oxygen is required in this reaction. So oxygen might come in anyway. So you can see in the next reaction, what happens? Further metabolism takes place in the mitochondria. So from here we will go into the mitochondria, right? Coproporphyrinogen is oxidized to protoporphyrinogen 3 by action of coproporphyrinogen oxidase. Okay. This enzyme specifically acts on type 3 series and not on type 1, and 2 propionic acid side chains are oxidatively decarboxylated to vinyl groups. Okay. And this reaction also requires molecular oxygen. So what happens? Oxygen is coming and CO2 is produced, and protoporphyrinogen is formed from coproporphyrinogen via action of coproporphyrinogen oxidase. Okay. Well, in this reaction oxygen is actually not required, only CO2 groups are removed. Here oxygen is required. We need oxygen in order to convert the propionic acid to vinyl group, whereas over here we can just remove carbon dioxide to form methyl groups. All right. So be very careful. The next step is actually generation of protoporphyrin. So from protoporphyrinogen it is getting converted to protoporphyrin, and this reaction also requires molecular oxygen. Okay. It is also happening in the mitochondria, and as per the discoverer, he named it protoporphyrin 9. And it is in this reaction that the methylene bridges are oxidized to methinyl bridges. Okay. There is a double bond incorporation over here. Okay. So we are getting protoporphyrin. We have almost reached the last step, because in the last step these 4, I told you that heme is actually ferroprotoporphyrin. So now iron, it needs to get inside. So iron by the action of the enzyme ferrochelatase or heme synthase is getting accommodated, and protoporphyrin forms heme. All right. So if we look into the valency of iron, it is actually attached to the 4 nitrogens. Okay. And the first atom, nitrogen atom of histidine, okay, 5 nitrogen atoms, and the remaining valency is satisfied with either water or oxygen atom, depending on the oxygen carrying, I mean, since the role is carrying capacity, I mean the role of iron is to carry oxygen in heme. So that valency, the last valency is satisfied by oxygen or water, right. So this is actually the entire pathway of heme synthesis, where all the intermediates have been shown. This is the number. So we started with 8, we ultimately needed 4. because 4 condenses into 1, which ultimately undergoes various changes to form a protoporphyrin, which by the action of the ferrochelatase enzyme incorporates an iron to become heme. Again, the name of the intermediates are necessary, the enzymes are necessary, but the structures are not. Only the final structure of heme is important, where we need to know the substituent groups. Okay. And if we look into this pathway linearly, this is how it happens. It starts from succinyl-CoA to glycine, this is the rate limiting enzyme, and thereafter all the reactions are taking place, and by the action of final ferrochelatase enzyme we are getting heme. We will again be